Hello, welcome to Biology, the scientific study of life. Insertions and deletions. Insertions and deletions are additions or losses of nucleotides in a gene. These mutations have a disastrous effect on the resulting protein more often than substitutions do. Because mRNA is read as a series of nucleotide triplets during translation, the insertion or deletion of nucleotides may alter the reading frame triplet grouping of the genetic message. Such a mutation, called a frame shift mutation, will occur whenever the number of nucleotides inserted or deleted is not a multiple of 3. All the nucleotides that are downstream of the deletion or insertion will be probably grouped into codons, and the result will be extensive missense, probably ending sooner or later in nonsense and premature termination, unless the frame shift is very near the end of the gene. It will produce a protein that is almost certain to be non-functional. Mutations. Mutations can arise in a number of ways. Errors during DNA replication, repair, or recombination can lead to base pair substitutions, insertions, or deletions, as well as to mutations affecting longer stretches of DNA. Mutations resulting from such errors are called spontaneous mutations. It is difficult to calculate the rate at which such mutations occur. Rough estimates have been made of the rate of mutations during DNA replication for both E. coli and eukaryotes, and the numbers are similar. About 1 nucleotide in every 10 to the power 10 is alterated and passed on the next generation of cells. A number of physical and chemical agents called mutagens interact with DNA in ways that cause mutations. In the 20 years, Hermann Muller discovered that X-rays cause genetic changes in fruit flies. With X-rays, he was able to make Drosophila mutants that he could use in his genetic studies. But he also recognized an alarming implication of his discovery. X-rays and other forms of high-energy radiation pose hazards to the genetic material of people as well as laboratory. Organisms. Mutagen agent radiation. A physical mutagen includes ultraviolet UV light, which can cause disruptive thiamine dimers in DNA. Notice this figure. Notice also base wall type mRNA, protein, amino acid, and carboxyl end. Notice that. Stop 3. Base pair insertion or deletion. Base pair insertion or deletion, strictly speaking, the example at the bottom is not a point mutation because it involves insertion or deletion of more than one nucleotide. Chemical mutagens fall into several categories. Base analogous are chemicals that are similar to normal DNA bases, but that pair incorrectly during DNA replication. Some other chemical mutagens interfere with correct DNA replication by inserting themselves into the DNA and distorting the double helix. Still, other mutagens cause chemical changes in bases that change their pairing properties. Researchers have developed various methods to test the mutagenic activity of different chemicals. A major application of these tests is the preliminary screening of chemicals to identify those that may cause cancer. This approach makes sense because most carcinogens, cancer-causing chemicals, are mutagenic, and conversely, most mutagens are carcinogenic. What's a gene? Revisiting the question. Our definition of a gene has evolved over the past few chapters as it has through the history of genetics. We began with the Mendelian concept of a gene as a discrete unit of inheritance that affects a phenotypic character. We saw that Morgan and his colleagues assigned such genes to specific loci on chromosomes. We went to view a gene as a region of a specific nucleotide sequence along the length of a DNA molecule. Finally, in this chapter, we have considered a functional definition of a gene as a DNA sequence coding specific polypeptide chain. All these definitions are useful depending on the context in which genes are being studied. Summarizes the path from gene to polypeptide in a eukaryotic cell. Even the one gene, one polypeptide model must be refined and applied selectively. Most eukaryotic genes contain non-coding segments, introns. So large portions of these genes have no corresponding segments in polypeptide. Molecular biologists also often include brometers and certain other regulatory regions of DNA within the boundaries of a gene. These DNA sequences are not transcribed, but they can be considered part of the functional gene because they must be present for transcription to occur. Our molecular definition of a gene must also be broad enough to include the DNA that is transcribed into rRNA, tRNA, and other RNAs that are not translated. These genes have no polypeptide products. Thus, we arrive at the following definition. A gene is a region of a DNA whose final product is either a polypeptide or an RNA molecule. For most genes, however, it is still useful to retain the one gene, one polypeptide idea. We have learned in this chapter in molecular terms how a typical gene is expressed by transcription into RNA and then translation into a polypeptide that forms a protein, a specific structure and function. Proteins in turn bring about an organism's observable phenotype. A summary of transcription, and this figure represents summary of transcription and translation in a eukaryotic cell. This diagram shows the path from one gene to one polypeptide. Keep in mind that each gene in the DNA can be transcribed repeatedly into many RNA molecules, and that each mRNA can be translated repeatedly to yield many polypeptide molecules. Also remember that the final products of some genes are not polypeptides but RNA molecules, including tRNA and rRNA in general. The steps of transcription and translation are similar to in prokaryotic and eukaryotic cells. The major differences is the occurrence of RNA processing in the eukaryotic nucleus. Other significant differences are found in the initiation stages of both transcription and translation, and in the termination of transcription. Notice here the steps also transcription. RNA is transcribed from a DNA template. Notice that DNA transcript, DNA. Stage 2 or step 2, notice RNA processing. In eukaryotes, the RNA transcript pre-mRNA is spliced and modified to produce mRNA, which moves from the nucleus to the cytoplasm. This is cytoplasm, nucleus or cytoplasm. 3 also notice that poly and cap, poly A and cap. Formation of initiation complex after leaving the nucleus. mRNA attaches to the ribosome. Notice that stage or mRNA. Notice that ribosomal subunits. Each amino acid attaches to its proper tRNA with the help of a specific enzyme and ATP. Notice also poly and cap. Notice anticodon, each one. Translation, succession of tRNAs at their amino acids to the polypeptide chain as the mRNA is moved through the ribosome. Notice one codon at a time. When completed, the polypeptide is released from the ribosome. Notice codon here, and this ribosome. Notice also anticodon A, E. Okay. Thanks for listening.